Hello, I would like to thank Cortigen for the great hospitality and the wonderful conference, of course, and all of the sponsors as well. My name is Mark Lewis, founded NAPRO in 2011, and that was the result of trying to do at-home R&D piggybacked upon service laboratories in California. I'd always known that, I had a fond appreciation for plants, so I'd always loved flavor and aroma and high oil content, whether it was basil or cannabis. And when I tried to do R&D in 2007, I had all these issues because I couldn't get any type of consistent results from lab to lab. So in 2011, NAPRO set out to accomplish several tasks and do them as quickly as possible. The first thing we wanted to do was create a robust and reliable, validated assay to look at and screen all of the relevant terpenes, and as well as some of the not so relevant or unknown ones. But we ended up testing for and validating for 17 and 11 cannabinoids. Then be able to communicate that data in a meaningful way, not only to patients, but also to plant breeders or physicians or just the connoisseur, someone who wants to learn more about the plant. During that time, we collected a bunch of data from 2011 to 2015, looking at essentially the whole chemoscape of what was popular medicine in California at the time. Once we had that data set, we did a few different analyses on it. The first analysis was to really analyze these cultivars, see what metabolites were present, and see if any patterns formed in potentially like a classification system based on secondary metabolites. The second analysis was to observe a single genotype over many, many replicates in the exact same environment to see if what the real metrics are for consistency and quality, to establish quality metrics for certain clients. Analysis 3, we wanted to compare homozygous hybrid seed with the asexual clonal propagation to see if the RSDs from plant to plant could be minimized versus the RSDs from lot to lot for the single genotype. The reason this is important is because, as you learn as a chemist, all systems can be defined by the summation of your inputs and outputs. So garbage in equals garbage out, and that's for us. If the medicine we're taking, we don't know what it is, you can't black box that. Or for a plant, if you're giving it stimulus, either environmental or chemical, you have to make sure you know what those are so that you don't get the garbage piece. In 2011, we were provided a unique opportunity. Someone had a series of controlled environment production facilities and 56 known cultivars by common name. These cultivars have been grown by these same cultivators for decades, so we knew that Blue Dream was Blue Dream and OG was OG, and of course I have quite an experience with the plant as well, so our team was able to match common name with the actual genotype that was being produced, which kind of flew in the face of some previous scientists who said, what's in a name? And essentially, there is a name game out there because the marketplace demands cookies. You have to have cookies. So if you don't have cookies, it suddenly becomes cookies when it moves across a state line. And the further you get across, the further the variation becomes. So the thought here was, well, let's figure this out. Let's look at how cultivating in the same environment over and over again affects a single genotype, and let's do this thing like real people, real scientists. So I'm gonna go through some of the data that we collected and explain why it was good data, and that's because we took painstaking efforts to ensure that the lighting was the same, the headspace above the light was the same, the rooms were the same dimension, the tables were the same, the soil was sourced from the same vendors, same techniques and all that stuff. And NAPRO oversaw the entire project. We collected climate metrics over the course of the entire lifespan of the plant, making sure to produce control charts, something that people in the industry often kind of have that starry, deer-eyed look when you say. so that if there were out-of-bounds situations, you could eliminate that from the data set as a flaw. Similarly, with the nutrient metrics, we monitored what went into the soil system, what went on the leaves, and made sure that it was consistent and the same across all batches. Even if we were spiking humic acid on day 40, it needed to be day 40 every time. And this is all part of the GE equals your phenotype chemotype equation that's shown there. So what we tried to do is minimize that delta E of part of the equation. And then that produced a phenotype, and we measured those metrics via tape measure: size of the bud, height of the flower, biomass, pretty standard. Took pictures along the way so that it looked nice when you went from page to page. I don't know what the pictures do other than that. But the next piece was a little bit more tricky. We had to actually collect the data from the chemotype stuff, right? So there wasn't any testing laboratory out there that was really doing terpenes at the time. Everyone was just dabbling in it a little bit. So what we sought out to do was develop this methodology. We paid to make it open source, so anyone starting a laboratory anywhere can download this guideline from sample prep to your analysis, and you can get reliable data for the cost of an HPLC and a GCFID. We answered a lot of tough questions: sample prep, extract stability. I mean, intraday, some of these matrices have degradation of certain terpenoids and formation of other ones, so you have to be really careful. It's all about the RSD. So just take a look at our material and data flow so you know that we thought about it a little bit. We look at a living plant, we took the morphological data, combined it with the environmental data, took a sample, followed it through the drying, curing process, collected that data. In this particular show, we're going to talk about the tier 1 assays we did, which is a single extraction in ethanol to create samples. We have to do one dilution for the cannabinoids. If you know, cannabinoids less than 2%, greater than 30% falls outside the line of Beer's law. So you have to do that dilution. A lot of people weren't doing dilutions in 2011. They were just shotgun blasting it. So we knew that there was RSDs associated on one side of the curve or the other, which could explain the conformational bias of the increasing amount of THC found in flowers through 2000 to 2015, because there was 36, well, that's crazy. So then we put that in the database, create an analysis, create our control charts, and then it says MCF sheets. That was a really old name for what we call a phytofacts report, a report that kind of gives you a flash of what that cultivar is. So to name a few of the 56 cultivars we examined in this, there's everything from Pincher's Creek, Old Amsterdam, classic Trainwreck, everything that you can imagine is on there. And that's Girl Scout cookies on the left, if you're curious, and Candyland on the right. I thought I'd make the presentation fun by putting pictures of buds up there, because we haven't seen any yet. It's all been really heavy, you know? And I forgot my joke. Oh my gosh. Thank you. You know, remember the rainbow at the very beginning? There's a greenhouse full of weed at the end of it. There really was. So the first thing we looked at was total cannabinoid concentration across the entire population of 56. This is kind of the distribution here. You know, we had a max of 36 or so percent cannabinoids and a min of 10 with, you know, an average of around 20, which kind of jives with what everyone's saying. Lot to lot RSDs, you know, average about 12%, and we found that the highly sampled cultivars had lot to lot RSDs of 10 to 20%, which kind of, you know, Arno Hoscamp's number of 15% falls right in the middle there. And we've seen some things have more variability and some things have less, and we just started to wonder, do the things that have less variability have less of an environmental impact, maybe more stable somehow with the environment? So when we went to look at terpenes, total terpene concentration ranged from about 0.5% to 2.5%, with a mean is about 1.5%, and this is in indoor under artificial lights, just to be clear. Lot-to-lot RSDs were increased for the terpenes, which kind of symbolized that the terpene biosynthetic pathways are more influenced by epigenetic factors. And overall, we found that cannabinoid content was more stable than terpenoid content between lot to lot. One of the things that this brought up was, well, is there a correlation between high terpene production and high cannabinoid production? And when we looked at that, we saw that there is indeed a correlation between higher essential oil production and higher cannabinoid production. However, the correlation factor is really low. That means that only about 15% of the factors influencing the terpene content derive from the cannabinoid content, which means that 85% of that is coming from somewhere else, whether it's genetic or environmental, we don't know. The correlation's real, just really tiny. But what does that tell us about each individual unique cultivar and how someone can use this tool to be able to treat specific ailments? Because, you know, everyone says, well, what's in a terpene? You know, Dustin started talking about linalool increasing efficacy. Mara talked about the entourage effect. Thanks to Dr. Machulam for coining the phrase. And it's like we have to look at each individual terpene. So we created this really, oops, I skipped a slide, forgot. Here's the whole spectrum of plants from the low concentration of THC to high, low terpenes to high. They're in 4-letter code, so it doesn't tell you a whole lot. I'm just showing you the spread. But the individual terpenes themselves we colored with color coordinated so that they would be intuitive, but also informative and create what we call a PhytoPrint. It's essentially the fingerprint of a particular plant. We started this with basil, and this slide actually had basil where the buds are now. And sometimes I think about if we could grow a bud that big, how that might be pretty incredible, especially after drying. Could you imagine how big it would be on the plant? It's crazy. But this is nice. So essentially, terpenoline is red, ocimene is orange, limonene is yellow, pinene is green, myrcene is purple, linalool lavender. So they're all intuitive. You can create collateral that, you know, color matches, and it shows different types of effects that these things do. But more importantly, you can look at different cultivars and compare them. And you have a couple examples here. But essentially, if you look at the major terpenes that are produced by the plant, they really class off pretty quickly. Either you have one major terpene, 2 or 3. It's more rare to have 4, 5, or 6 all equal amounts. So if you look at Bubba Kush, East Coast Sour Diesel, and Crypto, they both have high amounts of limonene and caryophyllene. However, you can see the myrcene and the linalool disappears in Crypto, which those subtle minor differences are how you differentiate the subtleties between each cultivar. Across the bottom, you have Classic Perps, Blue Dream 2, and it's just 2 because there are multiple cups of Blue Dream, which I'm sure a quarter knows about. And then Old Amsterdam, just a different purple class. We call this purple class because before we put science behind it, we used our indigenous knowledge. All the purples tended to be myrcene dominant, but we just called them purples because they're purple, and then it turned out that it worked out in our color scheme, thank God. That would have been problematic if it didn't. But you can see the subtle differences here in the pinene and the caryophyllene between the different cultivars, and that's how you really differentiate each cultivar. But they're all really within, these 3 cultivars are across 2 classes. If we look at some archetypes, and these are, there's a terpenoline dominant, that's associated with like your Trainwreck, limonene dominant. This particular is an OG, it's a OG Legend. Myrcene dominant, that's purple. Anything that was CBD prior to 2014 was pretty much all myrcene dominant. Ocimene dominant, this is a Hawaiian plant. I mean, some of these things produce like 2% of a single terpenoid. Pinene dominance, beta-caryophyllene dominance. And terpenes are important because beta-caryophyllene actually is a CB2 agonist. So if you're only testing for cannabinoids and you're giving your child a preparation, you should be very cognizant to the fact that when you concentrate this, CO2, anything, you lose your monoterps, but you usually get a ton of the caryophyllines, whether it's alpha or beta. They're present, so you should be aware of that. And if you hybridize 2 of these, like the pinene group and the myrcene group, you'll get something that kind of looks in between the 2 or a combination of the 2. This is interesting because, you create custom medicines this way, right? And if you could just buy a seed that was the medicine you wanted instead of, what would you call it, the lottery? You don't know what you're going to get. It's like a box of chocolates. So when we do a dendrogram of this chemical data, you get a nice breakup of hierarchy. We call it the terpene superclass system. It's just based on metabolite. So if pinene is in the major terpene world, then those cultivars fall in that family branch. Limonene, caryophyllene as such, myrcene, ocimene, and terpenoline. Interestingly, limonene and caryophyllene seem to be quite coupled, and ocimene and myrcene quite coupled in the plant. So lots of neat trends you see here, but just showing some stuff. And from that bore the classification system, where we wanted to do away with indica and sativa, because we don't really care about 20 years ago or 100,000 years ago. We care about right now and what it's going to tell our patients. So if you use a simple alpha system here to outline what the top 3 terpenes are, in this case it's limonene, caryophyllene, and myrcene for LCM. 1 means it's a type 1 plant, and G means that cannabigerolic acid is the second most abundant cannabinoid. Then you have a quick cartridge, a 5-digit item code that would work in a QR system for inventorying at dispensaries and totally removes the common name game. This led to having to do a PCA analysis on the data points to make sure that our classification system would hold water. And as you can see, it did. The ellipses are really great clustering for different chemotypes. If you notice the purple here, the blue dreams, there's also Kin's GDP thrown in here, and there's actually 3 cuts of blue dream. Then you have headband and several different OGs. You have purple Trainwreck and classic Trainwreck, and then so it's not just the same genotype. Well, it may be based on the world, but we know that the Trainwreck ones aren't because one was purple and one was not. So the point is that this system works. However, some genotypes have too much lot-to-lot variability, and over the course of, say, 40 or 80 runs, you have this plant that slides from 1 or 2 different classes, right? In this case, we're looking at OG Kush, another great picture of a wonderful flower. It's an MCL1T class, as we designate it internally. That's myrcene, caryophyllene, limonene. These things have 2% terps always and greater than 25% cannabinoids. It's truly a beast. OGs are marked by equal amounts of limonene, caryophyllene, and myrcene quite regularly. But if you look at these, this PhytoPrint over, say, a dozen or so runs, you have a separation of a couple different classes here, where sometimes the limonene's dominant, sometimes the caryophyllene dominant, and then you have this myrcene dominance across here. All of this is highly contingent upon environment and time of harvest, of course, and post-harvest conditioning. So you can have terpenes degrade to other things. So the point here is that quality control and laboratory integration with production is absolutely essential. And the idea of having third-party laboratory services that you send samples to is kind of like, to me, because it doesn't make sense unless you're just making pure extracts. And then you're still waiting in a holding pattern for your results, and not even guaranteed if they're correct. And if you have to send it to 3 different laboratories, oh my gosh, and then how do you pick which one? Do you average them? I don't think so. So it's complicated. Especially when, this is the same genotype, same environment over and over. If we look at a control chart here for total terpene content and total cannabinoid content, we ran a little experiment just trying to check out, I'm trying to speed up here, check out the difference between organic practices plus biostimulants, which biostimulants are that biome thing that Kevin was talking about at the beginning. Well, if we take away the biostimulants and just do organic practices, we see a significant decrease in both terpenes production and cannabinoid production. The producers didn't like that very much, so we had to quickly change. That's why there's only a few data sets there. And then we went to conventional agriculture, moved to salts away from organic practices, and brought the biostimulant back. And we got a boost in THC compared to the baseline and a decrease in terpenes. So it was very interesting for us to it confirmed what the grower said all along. They had to do it this way to get the oiliest, tastiest products, but at the same time, there's a neat way to boost your THC by switching to salts. This variability in cannabis brings up the quality question. The industry seriously lacks QA, QC. Most producers don't even have established quality metrics. I mean, it's like you have to have size of if it's a flower, you have to have size, shape, color, all these things most people don't consider. The only seed on the market you can buy that's true breeding currently is cannabis seed, and it may not even be in most cases. Unlike corn, you can't just buy a disease-resistant OG or a bicolored Kush. But people are making advances, and one of our clients wanted to make a type 2 Trainwreck because this particular person had anxiety, and she felt that Jack Herrera really made her, feel calm, and she wanted a type 2 version. This Trainwreck is really a TXX1G class, which means it's terpenoline dominant type 1 with cannabidiolic acid as the second most abundant cannabinoid. That's a picture of the classic Trainwreck right there. Very, very specific morphology. When you look at total cannabinoid concentration across all the individuals in the class, it's really nice and homozygous, all at about 1 to 2. I was going to say that we do everything exactly backwards. We'll start from the plant, you gotta start from the humans say CBD to THC, and the plant breeders say THC to CBD. So, such is life. But if you look at the terpenes, it's beautiful. They're all in the same class. In this particular data set, there was like 70 individuals, but I didn't want to clutter the screen with PhytoPrints. The point is that things are changing in the world of propagation, and we should start thinking about this because the RSDs from plant to plant here were less than the RSDs of a single cultivar from lot to lot. This begs up the question of indica versus sativa again. I don't know. I think it might be better used as an effects gauge than a genealogical nomenclature in the current system of medicine, not taxonomy. So end on a set of sample reports that I was talking about before, where you have the top terpenes color-coded at the top and the bottom to give you a feeling of the plant, total cannabinoids, terps, and moisture content along with the class, a picture of the flowers so you can visually identify the product tested with the product on the shelf. We have suggested entourage effects that are based on algorithms and patient panels done over the course of 2011 to 2015. Little flavor, spider graph, just a really cool way to do it. Warm tones are typically more stimulating, cool tones are typically more sedating, so it's a very intuitive system. So that's how you have it. Thanks.